Hi everyone, my name is Sua Lee from the University of Illinois, and we are going to discuss how the energy and nutrient digestibility are changed when dietary starch increases and dietary fiber decreases in diet fed to growing pigs. Equipment to determine the energy values of feed ingredients is available only at a few universities in the world, but the University of Illinois recently constructed such a facility. This facility has a capacity to hold the multiple number of pigs depending on the size and is installed with indirect calorimeter chambers that are airtight, which enables us to measure the gas consumptions and productions of pigs. In addition, because pigs are group housed, pigs share one feeder and move around, which may impact the energy expenditure. As you can see here in the cartoon, the fecal and urine pens are installed inside the chamber, which enables us to collect feces and urine separately. Therefore, using the system, concentrations of digestible energy, metabolizable energy, and energy and digestibility and retention of nutrients can be determined in diets fed to pigs on an ad libitum basis. And because the way pigs are housed is similar to the way pigs are housed in commercial settings, this system is believed to generate values that are representative of commercial settings. Using this system, accurate prediction of net energy in diet fed to group house pigs requires an understanding of how each nutrient class influences energy utilization and nutrient digestibility. Therefore, we have conducted a series of experiments to determine net energy and nutrient digestibility in diets containing a wide range of carbohydrates, lipids, and protein to develop a new NE system ultimately. Among the energy containing nutrients, let's talk about carbohydrate this time. Starch is a highly digestible carbohydrate source, whereas fiber is associated with lower digestibility and increased energy losses in feces, urine, or thermal heat. Therefore, an experiment was conducted to test the hypothesis that increasing dietary starch and decreasing dietary fiber increase net energy and nitrogen balance and the digestibility of nutrients. There were 6 experimental diets. The first 5 diets contained corn DDGS from 35% all the way down to 7%. Corn was used as the major feed ingredient in place of DDGS. To generate a high starch diet, 10% cornstarch was used in the last diet. Because corn has more starch and less fiber than DDGS, it was expected that concentrations of starch increases and concentrations of fiber decreased in these diets. Indeed, the analyzed starch was 30% in the first diet and increased up to 56% in the last diet, and the analyzed total dietary fiber was 21% in the first diet and decreased to 12% in the last diet. We used 2 groups of 24 pigs, and 4 pigs were housed in a chamber. In each group, the 6 chambers were allotted to 6 treatments using an incomplete 6x3. Because there were 2 groups, there were a total of 6 replicates per treatment. Pigs had ad libitum access to feed and water. After 7 days of adaptation, feces and urine were collected for 6 days using the time-to-time procedure. During the collection period, consumption of oxygen and productions of carbon dioxide and methane were also measured to determine the total heat production. Diet and fecal samples were analyzed for dry matter, gross energy, nitrogen, total dietary fiber, and ash. Urine samples were analyzed for gross energy and nitrogen. Using the analyzed values and weights of feces in urine, the apparent total tract digestibility (ATDD) of nutrients and energy and retention of nitrogen were calculated. Concentrations of digestible energy, metabolizable and net energy were also calculated. The statistical model included diet as fixed variable and group period and chamber as random variables. Contrast coefficients were used to determine linear and quadratic effects of increasing starch and decreasing fiber. Significance was set at alpha less than 0.05 and tendency at 0.10. Moving to the results, let me set up the slides first. To better communicate, the inclusion rate of DDGS was used here, which ranged from 35 to 0%. The research indicated that the ATDD of dry matter and gross energy was increased as DDGs decreased and starch increased in diets. Now we are looking at nitrogen balance data, and the research indicated that the apparent thoracic digestibility of nitrogen and retention of nitrogen increased by DDGS and increasing starch in diets. The ATDD of total dietary fiber also increased as DDGs decrease and starch increase in diet. Likewise, the ATDD of ash was increased by decreasing DDGs in the diet. Respiratory quotient was not affected by dietary treatment, but net energy tended to increase when dietary DDGs was reduced and starch was increased. We also conducted another experiment to determine the apparent ileal digestibility of a dry matter, starch, and amino acids to demonstrate the relationship between net energy and nutrient digestibility in the same diets. Data indicated that the apparent ileal digestibility of dry matter and starch increased as DDGs decreased and starch increased in diets. The ileal digestibility of acid hydrolyzed ether extract was also increased by decreasing DDGs in diet. Same was observed for standardized ileal digestibility of a total amino acid and other amino acids. The observed increases in digestibility of a dry matter, gross energy, and acid hydrolyzed ether extract with decreasing DDGs inclusion aligned with previous data. The observation that NE was increased by decreasing DDGs also agreed with the understanding that diets with low fiber and high starch have increased energy utilization because starch is used more efficiently than fiber. These data will be used to establish a new NE system. Therefore, further research is needed to determine the NE and nutrient digestibility in diets containing a wide range of protein and fat. Thank you for listening to my presentation, and I would like to acknowledge everyone from Dr. Stein's lab. If you want to learn more about our research that we are conducting in the Stein Monogastric Nutrition Laboratory, please visit our website. Thank you.